It's utterly fascinating because, again, as you said, it's a pattern that is playing out on the level of the branches of one dendrite in one single neuron, so at the single cell level, at the vascular level, at the pulmonary level. And it's the same thing that's happening with a tree, with its literal branches. And it's the same thing when you look at a satellite photo of the Nile Delta as it empties into the Mediterranean, splitting into branches like that. And one thing that's clear is you can't possibly code for that system with your genes in a point-for-point way. Simple example, just to take a neuron will have about 10,000 dendritic spines, meaning about 10,000 branch points in the course of it snaking outward. And you cannot have each branch point specified by a single gene, because we only have about 20,000 genes in our entire genome. You can't be using up half of it just on like the branching in one cell type. So the challenge is, how do you get branching? How do you get this repeating pattern in a scale-free way without having to use too many instructions, especially genetic instructions, that could then play out on the level of single cells and entire organ systems? And what you get are rules that have to emerge from the biophysical traits of the system, which is to say you have to have some sort of rule which will be applicable whether you are talking about the branches on a dendrite of a single cell or pulmonary branching involving thousands, millions of cells. And what you have to have are laws to the effect of, say, if you have a tube, a dendritic branch, a tube, a blood vessel, a tube, the pulmonary system, you have to have some sort of rule of a consistent relationship between the diameter of the tube and the length that it grows. And you can have a rule like whatever the diameter is, grow 5 times that length and then split in 2. And at that point, each lumen is half the diameter of the original one. So growing 5 times the length is 1/2 the length, and then it splits again and splits again. So that's great on sort of a informational level, mathematical level of just when do you know how to bifurcate? How do you make that as something that could play out in a single cell and then in entire organ systems? And it's much easier to describe visually, but there's some fantastic models out there for how you get, say, basically the tip of a growing branch, which will have some sort of growth stimulating factor on top. And a critical feature of it is, as it presses up against that pocket of growth factor, by pressing up, it splits it in 2, each half the size of the original, which now triggers growth in that direction, which when those reach the top, split each of those in 2, which now has it branch out and branch out. And in each case, you were literally taking a volume of growth encouraging factor, and by splitting it in 2 to either side, the next branch will be half the distance before it splits that in 2 and half the distance. And these are theoretical models. Amazingly, John von Neumann, who's sort of the founder of cellular automata and computer science, he's the one who actually came up with this model in the 1950s. How could you explain branching patterns in nature? And he made some very explicit predictions about you'd have to have a growth stimulating factor with this or that property, and a growth inhibiting factor with a different sort of property. And in the last few years, people have gone and looked, and that's exactly what you find in the developing lungs, in the developing vascular system, and in neurons doing branching. So it's rules that have to be scale-free, and thus we are looking at a fractal biology in these cases.